So I'm interested in applying home cage monitoring to biomedical studies, and I did a short investigation of how IntelliCage system is used for drug development process. And for the purpose of this talk, I decided to focus on this unique opportunity that IntelliCage gives, which is that we can perform longitudinal studies. So we can observe mouse behavior over a prolonged period of time, which is especially useful for monitoring age-dependent diseases like neurodegenerative disorders, which have been already tested and described on different models. As an example, it was Huntington's disease, investigated by at least 2 groups, and Alzheimer disease, also described over a long period in IntelliCage. And there was also a recommendation to include IntelliCage system to preclinical studies of amyotrophic lateral sclerosis. It was a postulate of one of the group working with this disease, but unfortunately it was only a recommendation. I haven't found any research paper connected with this disease. So if we take into consideration a long-term studies in IntelliCage, we can also test drug addiction, and for example, alcohol addiction was very well investigated, and we can distinguish different phases of this addiction. And if we think about therapeutic intervention, I believe it would be possible when we know the process of this alcohol addiction. Also, other substances were given and tested for a longer time in IntelliCage, and more examples can be found in an excellent review of Iman from last year. And there is another aspect of this longitudinal studies, which is developmental neurotoxicity. So we don't focus only on gene influence on behavior, but also we can look at early life exposure to environmental factors, like, for example, chemicals that can influence the later stages of life. So we can look at the epigenetic factors. So, for example, given the chemical compounds to pregnant mothers and test later their F1 generation. And also from the point of view of drug development, it is obligatory to perform developmental and reproductive toxicology in one species. And I believe that IntelliCage can be used for that. I haven't found such a publication yet, but I believe that it is possible to some extent, of course, in mice or in rats only. So when we have a mouse model of the neurodegenerative disease, so first thing that when we think about preclinical testing, we have to decide, we have to find out what is the disease progression, how the disease develops during age. And there are several approaches made in IntelliCage how to check that. So the group of Balci tested 2 strains of mice. With the first one, R62, had a short fragment of mutant human Huntington inserted, and it was their approach was to introduced the mice to IntelliCage at the age of 7 weeks, and then they were tested with the same schedule over and over for 4 weeks. So they started when they were 7 and finished when they were almost 11 weeks old. And the schedule was the first habituation with all corners open, so mice could recognize that corners are available and they can drink there. Then they switched to alternation task, in which mice could drink only from 2 corners and in a special order. So when they drank from corner 1 and only from bottle on the right, they had to go to the corner 2 and could drink from the bottle on the left. So that was a I would say, quite a difficult task. And even if they had
Sorry to interrupt, but I just want to make sure we still can see the first slide. Is it fine?
Oh, sorry. I don't know why. Okay. Sorry.
Okay, it works.
Yeah. So yes. So maybe I will come back shortly here. So we had here the division of 3 different types of studies that can be performed in IntelliCage, longitudinal studies. And now I'll try to. Okay, so in this paper, which I was describing, there are 2 genetic models of Huntington's disease, and the first mice were tested starting from the age of 7 weeks, and they were tested 4 times with the same schedule. So first it was habituation, then it was alternation, which was rather difficult. And authors wrote that even if the paradigm was repeated few times, some mice couldn't learn, they didn't drink, and after few tries, they had to remove them from the cage. So that was the approach when the mice were tested over and over to see if there will be any changes when the disease progressed. But the results were not very informative. So during such a short time, it was difficult to find a change. But the other strain of mice, which expressed full-length human mutant Huntington gene, was tested in a different way. So the mice were divided for 4 groups, and they were tested when they were 16 week old, 24, 36, and 52. So there were 4 cohorts, and they were introduced to IntelliCage independently, and they were tested for 16 weeks. So this approach can show us how so we have a longer period of time, and we can see at least a kind of dynamic of changes. And also, if we introduce different age groups independently, they are a kind of naive mice. So we have a clear situation how the first time learning look in these mice. So that were two different approaches for long-term monitoring, and in other study of Alzheimer disease model, there was another approach. So mice were mutant mice with the humanized APP knock-in carrying Swedish, Iberian, and Arctic mutations. There were different groups with 1 or 2 or 3 mutations. And in this study, the authors from Japan, they perform a series of experiments, and they performed them twice. So on the same mice, they started when mice were 8 months old, and then they introduced them to IntelliCage, and they had a place preference task in which they have one corner correct from which they can only drink. Then it was reversal to other corner. Then in the next step, one of the corner was punished, so they received air puff when they wanted to try to drink. And then that was a more difficult task D, which was serial reaction time to evaluate impulsivity and attention control. So in this task, animals first had to wait with a delay of 2 seconds and then react quickly after the LED flash. So that was quite challenging task. And the last one was delay discontinuing, which was already mentioned by David. So this task did evaluate compulsivity, in which animal can choose between immediate access, water access, or delayed, but receive saccharin, which is more appetitive liquid. Yes, so in this approach, the same mice were tested twice, when they were younger and then when they were older. Authors also introduced these mice for the third time when they were old, like 18 months old, but they don't show the results from this, the third stage. So this approach is also can be used when we want to check the dynamic of changes with age on the same animal. So I can see some potential therapeutic intervention here when we have described how the disease is changing. And what was also important here, that the animals were tested at the same time. So they had 8 IntelliCages, and in one it was Y-type mice. In the second, it was with 1 mutation, 2 mutations. In the third, 4 mutations. So the point here is that it is important also when we do the drug testing, that we usually, for example, for toxicity studies, the whole cohort of animals is tested at the same time, because we want to avoid these seasonal changes. We want to avoid temperature, different humidity, and so on. So, but of course, this is a limitation. Not all have 8 IntelliCage to check so many cohorts at once. So, so another limitation that I can see is that is a group housing. This is from one side an advantage of IntelliCage, but also it can be disadvantage, especially that in preclinical studies, both sexes should be tested. So either new drugs should be tested on males and females, and there are many studies showing that if we keep males together from when they are young, then we can move them to the IntelliCage, and they will be not very aggressive, but the situation may change if we give them a kind of any kind of drug. So so this is one of the potential obstacle. Another is that group housing may induce social ranking. So to prevent that, we can just check that if we have an outlier, which, or if we have a mouse which is I don't know, visiting corner as first when the corner was closed and now is open, then we maybe can observe this mouse and eventually remove it. And also it may influence behavior and task-related performances, but still we can kind of control it by putting a proper number of mice to one cage. So if we, I would say, if we have a group of more than 14 mice, we may have this group relations that we don't want to have, unless we are looking for social behavior or competitiveness or other tests. So there are some papers showing that genotype-based housing is better. It allows to avoid possible dominance of wild-type mice over transgenic or mutant mice, and it also allows to avoid disturbing wild-type behavior. Sorry for that. For example, sleep patterns. So I would like to show the results that we had in our lab. That was a study of APP mice with a single London mutation. That mice were well characterized behaviorally, and we tested 3 different age groups: 5 months old, 12 months old, and 18 months old mice. And what we found out when we employed group learning, in which in one corner there was a sweet water, and in 3 other corners it was a plain water, the mice had to, they learn that where is the location of the sweet water, and because they prefer it. And so the group of mice had to cope with the same task. And we tested 2 housing conditions. In first, mice were mixed in terms of genotype. So in one cage, both wild-type and APP mice were kept together. And in separated conditions, wild-type mice were kept separately, and transgenic mice, APP mice, were kept separately in independent cage. And what we found out is that when we had transgenic mice separated, they were not able to remember the location of the corner with sweet water. So their performance was about 30%, 25 to 30% of correct responses. But when we mix them with the wild-type mice, they were able to learn the position of this corner, and they learn efficiently. So this is the argument that genotype-based housing is worth considering, or at least comparing how these strains behave when they are separate and when they are mixed. Okay, and when we come back to this paper of Masuda, it was the experiment that was repeated on the same mice. And what the results show that on the left side, we can see that when the mice were younger, there were almost no difference in place preference and place reversal. It's A and B between groups and between genotypes. But when they were introduced to IntelliCage for the second time, there were still no difference in place preference, but there was a difference in place reversal. And similarly with the accuracy in attention test, in the first time there were also no difference, but when they were introduced when they were older, there were some differences. So they did not, either they did not remember the task, or they forget, or they were not able to recall this memory anymore. And also with delay discontinuing, here there were some differences when they were introduced for the first time, and they were even more prominent when they were introduced for the second time. I'm telling that because of the paper which was recently published. It was also from Japan, but other group with a company also included. And these results were reanalyzed using machine learning, exactly a deep neural network. So they took the results of Masuda paper, and they rank it. So they make parameters from each behavior, and these behavior were an input to a deep neural network algorithm. And according to this algorithm, they classified mice with a high risk of AD at the presymptomatic stage. So when they were first time introduced to IntelliCage, they found out that some of these mice were already in the AD risk, identified on the basis of compulsive and learning behaviors with about 90% of accuracy. And they claim that already in this early presymptomatic stage, using this algorithm, we can treat some of the behaviors as biomarkers for early screening. So this is, for us, it's also a good news that we gather a lot of data, and maybe we are not able to analyze them now, but maybe we will be able to make some effort in the future to produce, I know, more comprehensive results. So in this case, biomedical application is not only drugs, but also a kind of biomarkers that can be based on behavior. And another limitation, the last one that I have on my mind as for now, is the route of administration. So even if we have a well-characterized line and we would like to apply a new drug, we don't want to remove the mice from the cage every day for injection. We don't want to give them oral gavage or something like that that would disturb their well-being in IntelliCage. So what we tried so far is delivering drug by osmotic pumps, which also is not a perfect way, because we should be very careful with implanting the pump to the proper place so the mice don't stuck in a corner. And we tried also with drug solution in water, which is a lot of drug is wasted because we have to change it regularly, and some of them are sensitive to light, so they have to be covered. The bottles have to be covered. So there is no perfect way to deliver a drug to mice in IntelliCage. I will go quickly to the last results. That was a successful implementation of osmotic pump tested in a Vöikar conflict paradigm that was adapted to IntelliCage by SAFI. And the Vöikar test is first mouse is water deprived, and then when it tried to drink, it received an air puff, but only after the first time. Another attempt of drink is allowed, so they are punished only first time when they try to drink. And SAFI removed the mice from IntelliCage and injected them with diazepam. I implanted osmotic pumps of Alzet that released another anxiolytic drug. I didn't change it. So I applied alprazolam, and other group received saline. And so in alprazolam treated mice came back to the same corner when they were punished very quickly. These are the white bars. So after a few minutes, they came back and drank. And black bars are white-eyed mice that did not come back for drinking for 1 hour. So I was able to implement these mini pumps and receive the same results as injections. And what is more important, we were able to repeat the same drug effect as SAFI. So coming to conclusions, I can see that the IntelliCage system may facilitate biomedical research in understanding the disease at different stages, in monitoring response to pharmacological intervention, including chronic drug administration, in social context of drug effect, and in improving repetitivity and reproducibility of behavioral data across labs. Thank you.